hydrolysis of the C3, yielding to C3H2O that binds to factor B. In the presence of factor B, will cleave into C3H2O capital B small b called C3 convertase, which is a fluid phase because it's attached to a water molecule, and BA that initiate more breaking down of C3 into C3A and C3B. This is C3B bound to microbial surface and bind to factor B that cleaves into BA and capital B small b. In the presence of factor B, lead to formation of a C3B capital B small b called C3 convertase, which is membrane bound, and also BA. Formation of a C3 convertase, a C3B capital B small b, requires a protein factor, which is factor BP, that creates more C3. This is called positive feedback loop. This leads to formation C5 convertase, C3B capital B small b, C3B that act on C5. It cleaves into C5A and C5B and continue the same reaction as classical pathway C6, 7, 8, 9 and end with cell lysis. The second pathway of the alternative pathway is the proper-gene activated pathway. The proper-gene may also serve to initiate the complement by binding to component of microbial membranes. This is the proper-gene bind to the cell wall of the bacteria, for example, and stabilize the binding of C3b to capital B small b, lead to complex formation, which is C3b P capital B small b complex. This is that act as C3 convertase in the presence of the factor D. The third pathway of the alternative, which is the protease activated pathway. The proteins factors involved in a blood clotting, such as thrombin and plasmin, are capable of generating both C3A and C5A. C3B binds to microbial cell wall and stabilized by C6. And this is the summary of the complement activation. This is the classical, this is the lectin, this is the alternative. The classical activation by the antigen-antibody immune complex lead, which is a rule, which is lead to complement activation, and the C1, C4, 2, 3, 5, 6, 7, 8, 9 lead to pores formation and cell lysis. And the lectin through the mannose binding lectin, which is act instead of C1QRS and continue the same pathway. And the alternative, which is the spontaneous hydrolysis or pathogenic surfaces, which is C3 and fluid phase, and then bind with the factor B, and then continue C5 convertase and end with the cell lysis. Biological effects of the complement activation, cell lysis. The cell lysis occurs through the complement activation, and pores formation lead to target cell lysis, and opsonization by this is the bacteria opsonized by C3b, which is the red one, and enhance phagocytosis, which is act as an opsonin. And another is the activation of inflammatory response, and this is the complement receptor, and lead to extravasation of the cells and degranulation of the cells. All these involved in the inflammation. And the another function is the clearance of the immune complex. And this is immune complex, which is antigen antibody immune complex present in the circulation. And this is will be with the C3b, and this lead to enhance the phagocytosis and remove the immune complex from the circulation. The complement, in order to achieve its function, is need receptors, and this is called receptors for the complement. For example, complement receptor 1, CR1, which is designated CD35, present on the macrophage neutrophils and bind to C3b, and the function clearance of the immune complexes and enhance the phagocytosis. Another receptor is complement receptor 3, which is present on the macrophage and the neutrophil, and binding to adhesion molecules facilitates the extravasation of the neutrophil and other immune cells. The complement system need regulation is not continue activation and continue work for a long time. Once the inflammation subsides, once the infection is removed, so the complement system will function will be terminated, and this termination of the function of the complement needs regulation, which is called regulation of the complement activity. And this achieved by a C1 inhibitor, which is a promotes dissociation of C1 components. Second, decay accelerating factor, which is a promotes decay of C3 convertase. Third, which is a protein S, prevents insertion of C3b67 into the host cell membrane. Complement deficiency. When there is a deficiency in the complement component, so there is lead to formation of disease. First, C3 deficiency. When there is complement 3 deficiency, leads to recurrent bacterial infection. When there is deficiency in C9, leads to increased Neisseria infection, whether gonococcal or meningococcal. C1 inhibitor deficiency leads to hereditary endoneuritic edema. So, when there is deficiency in the complement component, lead to development of diseases. And thank you.